Hello everybody, this is D. Rajashekar, lecturer in Botany, Srimati Gangineni Kalyani, Government Degree College, Vinikonda. I welcome you all to this video lecture, C4 cycle. Before entering into this C4 cycle, just we will go back to the important details that were mentioned and covered in the previous videos. Photosynthesis is a unique and very broad metabolic activity that is only seen in plants. It is classified into two phases, light phase and dark phase. Light phase means conversion of solar energy into chemical energy, that is ATP and NADPH2. Dark phase means conversion of this chemical energy into potential energy, means conversion of ATP and NADPH2 into carbohydrates. Dark phase can be generally carried out by Calvin cycle, and the plants which perform this Calvin cycle on the dark phase are called C3 plants, because in Calvin cycle the first stable compound formed is PGA, that is a 3 carbon molecule. Broadly it can be called as phosphoglyceric acid. Up to 1965, everybody thought that plants perform dark phase only by means of Calvin cycle or C3 cycle. But later in 1965, Kortschok, Hart and Burr, these three scientists identified the formation of 4 carbon compounds as first stable compounds in the dark phase of photosynthesis. Especially these observations were made in certain tropical plants. Later in 1966, the other two scientists by name Hatch and Slack discovered the same facts in sugarcane. That is why this cycle is also called as Hatch-Slack cycle. Since it is alternative to Calvin cycle, it is also called beta carboxylation pathway, as the first stable compounds are carbon 4 compound, there it is also called C4 cycle. So far 11 families, 19 genera and 19 species have been identified, which are popularly known as C4 plants. The plants which perform dark phase by this Hatch-Slack or C4 cycle are called C4 plants. And the characters which make them separate from C3 plants can be called together as C4 syndrome. What are those characters which make C4 plants different from C3 plants? Are generally tropical plants perform the C4 cycle or Hatch-Slack cycle. This C4 cycle can be started even in the less amounts of CO2. Plants which do not have enough water can also easily perform this process, and morphologically, anatomically also these plants are different from C3 plants. What are the morphological features? How can you identify just by the morphology? Mainly in many of the plants, when you observe the veins, they are translucent and light in color. But in C4 plants, if you observe properly, the veins are green in color, because in the veins also we can see chloroplastids, especially in the bundle sheath region. In C3 plants, we cannot see chloroplasts in bundle sheath. That means in C3 plants we can see chloroplastids only in the mesophyll tissues, but in C4 plants chloroplasts are seen in both the mesophyll tissues as well as the bundle sheath cells. And anatomically, if you observe C4 plants, we can see, the morphology or anatomical features that are seen, that are shown in this slide. If you observe properly, here, this is the upper epidermis region and this is the lower epidermis region of the leaf, and all this tissue is called mesophyll tissue. Here in mesophylls you can see small dot-like structures, they are actually chloroplastids. Here in the center region we can see vesicular bundle. It is enclosed by a chain-like cells that is called bundle sheath. And if you observe in the bundle sheath cells, you can see oval shaped structures which are more larger and heavier than that of the dot-like structures that are already seen in mesophyll cells. So these are also chloroplastids. So here in mesophyll cells as well as in the bundle sheath cells you can see the chloroplastids. And you can also see the difference between the chloroplastids. In mesophyll cells, the chloroplastids are somewhat smaller than that of the bundle sheath cells. In bundle sheath cells, you can see somewhat larger chloroplastids than that of the mesophyll cells. And another important thing is, in mesophyll chloroplastids, we can see highly developed lamella and stroma regions. Whereas in bundle sheath cells, though the chloroplasts are larger in size, here the lamellar region is not well developed, that is called rudimentary lamella, whereas the stroma is a developed one. So having two types of chloroplastids by the same plant can be called as chloroplast dimorphism. Actually, as here the bundle sheath is like a chain or a wreath, the so-called anatomy of C4 leaf is called Kron's anatomy. Kronz is a German word which means a chain. So the advantage of C4 plants over C3 plants is C4 plants can withstand to high temperatures and they can withstand to less amounts of water. They can carry out or start this process even in the less amounts of carbon dioxide. And moreover, here we can see partition in the dark phase. Actually, in C3 plants, dark phase is carried out in the name of C3 cycle or Calvin cycle. But here in C4 plants, we can see two cycles, both C4 cycle as well as C3 cycle. C4 cycle is generally carried by mesophyll chloroplastids and as well as bundle sheath chloroplastids, but much part of C4 cycle is done in mesophyll chloroplastids, whereas C3 cycle is generally taken place in the chloroplasts of bundle sheath. Thus, the work is shared between the mesophyll chloroplastids as well as the bundle sheath chloroplastids. Because of this sharing of work is also one of the advantage for C4 plants for maximum photosynthetic activity. Thus, another difference between C3 plants and C4 plants is dimorphism in chloroplastids, and in C3 plants we can see only one type of cycle, that is C3 cycle, but in C4 plants we can see both C4 cycle as well as the C3 cycle, which are not done at the same point or same organelles or same region, but they are carried out in different regions, but in chloroplasts. Now we will see the total process of C4 cycle. As I have told you just now, here in C4 plants we can see two types of chloroplastids. This is a chloroplastid of mesophyll cells, and the below one, and this is called the chloroplastid of bundle sheath cells. See the difference. The chloroplast of mesophyll is somewhat smaller, whereas the chloroplast of bundle sheath is somewhat larger. Both are connected by plasmodesmata connection. Here you can see some gaps between these two types of chloroplastids. in the cells, they are called what? Plasmodesmatic connection. As I told previously, in both mesophyll cell and medullary sheath cell, C4 cycle take place, but C3 cycle or Calvin cycle takes place only in the bundle sheath cell. Here, first what happens? CO2 present in the atmosphere enters into the mesophyll cells of the C4 plants. The CO2 is first accepted by PEP molecules. They are 3 carbon compounds, phosphoenol pyruvic acids. By taking the CO2, PEP is changed into oxaloacetic acid, a 4 carbon compound in the presence of an enzyme PEP carboxylase. This is the first reaction of the C4 cycle. The first CO2 acceptor is phosphoenol pyruvic acid, shortly known as PEP, and the first enzyme is PEP carboxylase, and the first formed compound is 4 carbon compound, that is oxaloacetic acid. One thing we should know here, actually in C3 cycle the first CO2 acceptor is RuBP and the first enzyme is Rubisco. It is a combination of both carboxylase and as well as the oxygenase. There is a competition between the carbon dioxide and oxygen for RuBP. But here in C4 plants, there is no such a competition, because the process actually starts with the PEP, which takes carbon dioxide from the atmosphere, and in the presence of PEP carboxylase only, it is the single enzyme which changes it into oxaloacetic acid, and then thus formed 4 carbon compound oxaloacetic acid is changed into another 4 carbon compound, that is malic acid. So here the enzyme is malic acid dehydrogenase. Here one NADPH2 is consumed. This is called second reaction, the conversion of oxaloacetic acid into malic acid. Both are 4 carbon compounds. Here an NADPH2 is consumed in the presence of an enzyme malic acid dehydrogenase. Thus formed 4 carbon compound malic acid enters into the bundle sheath region. So first 2 reactions took place in the mesophyll cell. In the third reaction, this 4 carbon compound malic acid enters into the bundle sheath cell. Here it is changed into 3 carbon compound pyruvic acid, as well as 1 carbon is removed in the form of carbon dioxide. Here the enzyme is decarboxylase as well as the dehydrogenase. Why it is called dehydrogenase? Here one NADH2 is formed. In the previous reaction one NADPH2 is consumed. Here one NADPH2 is formed, so that the net loss and gain is equalized, and thus formed CO2 is utilized in the Calvin cycle. So after the 3 reactions in C4 cycle, the resultant CO2 enters into the Calvin cycle. As there is no oxygen, there is no chance of competition between CO2 and oxygen. So in the Calvin cycle, the first CO2 acceptor, that is RuBP, and the first enzyme Rubisco, where carboxylase is activated because it gets CO2 only. There is no competition between the carbon dioxide and oxygen. So that Calvin cycle takes place and sugar is released. This is the final product of photosynthesis. It is entered into the vesicular tissues region, that is xylem. Thus glucose or sugar is synthesized by C3 cycle only. And the pyruvic acid, the 3 carbon compound, enters back into the mesophyll region, where it consumes ATP molecules in the presence of kinase. Again it forms the first CO2 acceptor, that is PEP, phosphoenol pyruvic acid. Thus 4 reactions take place in C4 cycle, and in between the third reaction and fourth reaction we can see the Calvin cycle, where sugar molecule is synthesized. Thus the C4 cycle is completed. Why C4 plants are more effective regarding photosynthetic activity, especially dark phase? That is the major thing we have to focus on. In C3 plants, if you observe, there is no chloroplast dimorphism, there is no two cycles. There is only one cycle, that is C3 cycle. C3 plants face a competition between CO2 and oxygen for the first enzyme and the first acceptor. They do not carry out dark phase in high temperatures, less water available regions, and less carbon dioxide available regions. Whereas C4 plants have all these provisions. What are they? In C4 plants we can see chronogenotomy, chloroplast dimorphism, occurrence of two cycles, that is C3 and C4 cycles, partition of two regions to carry out two cycles separately, no competition between CO2 and oxygen for RUBP, and moreover C4 cycle occurs just below the surface region of the leaf, whereas the C3 cycle takes place in interior cells such as bundle sheath cells. There is no entry of oxygen at all. The CO2 that is liberated directly from C4 cycle enters into the Calvin cycle without delay, without competition. It immediately starts Calvin cycle and forms glucose molecules or sugar molecules. So, what is the total consumption of energy? Since photosynthesis is an anabolic process, it requires energy in the form of ATP and NADPH2. We all know that C3 cycle requires 18 ATP and 12 NADPH2 for the complete glucose formation. In C4 plants, as the C4 cycle and C3 cycle take place together, here the total requirement of ATP and NADPH2 are 30 ATPs and 12 NADPH2s. How 18 ATP and 12 NADPH2 in the C3 cycle and remaining 12 ATP are consumed in the C4 cycle, especially in the last reaction, that is fourth reaction, the conversion of pyruvic acid into PEP requires ATP. So, if for the total formation, for the formation of glucose molecule completely, for reduction of 6 carbons in the glucose, C4 plants need 30 ATP and 12 NADPH2. If one carbon is considered, then how many ATP and NADPH2 are required? For one carbon molecule reduction or one cycle, 5 ATP and 2 NADPH2 are needed. For 6 carbon reduction, as we all know, glucose is 6 carbons. It requires 30 ATP and 12 NADPH2. In 13 ATP, 18 are consumed in Calvin cycle and 12 ATP are consumed in C4 cycle. I hope you have understood C4 cycle clearly. Actually, in this video lecture, we have learned what is C4 cycle, how it is being called by other names like beta carboxylation, Hatch-Slack cycle, what are C4 plants, what is chronogenotomy, what is chloroplast dimorphism, how C4 plants carry out both C4 cycle and C3 cycle, how glucose is synthesized. In another video lecture, we will learn other details of photosynthesis. Till then, bye. Thank you.